students, welcome to BIOMICS HUB. Do you know how does a plant respire or how the exchange of gases takes place in the plant without any organelles, or how does the transpiration is taking place? It is due to the presence of a small opening present in the leaf surface of the plant, which is known as stomata. So in this today's video, we are going to discuss about the regulation of the stomata, that is how it is being opened and how it is being closed. So before that, we have to understand the structure of the stomata, structure of the leaf which are present. Like in the leaves, there is two layers which are present in the upper region and the lower region, which is known as cuticle. And this cuticle which is present, it is present for protecting the plant from excess loss of water. And below this is the epidermal cell which are present. In the upper region there is upper epidermal cell, and in the lower region there is lower epidermal shell, and in the between is a mesophyll cell. Okay, so near the epidermis there is minute opening of the cells. This is known as stomatal pore, or it is known as stomatal opening, where the gases exchange as well as transpiration take place. So this portion which is present, it is being regulated by some of the epidermal shell. One of them is the guard shell and the subsidiary shell. Here we can see the opening of stomata. How this opening of stomata and closing of stomata is taking place, we will discuss. So before that, let us see the structure. Here this outer layer which is present, it is known as the subsidiary shell. It is known as the subsidiary shell, and in the subsidiary shell is also the nucleus which is present. And this bean shaped structure which is present, it is known as the guard shell. This is known as the guard shell. And in case of dicots, the guard shell which is present, it is of kidney shaped or bean shaped. Okay, and in case of monocots, it is of dumbbell shaped. It is of dumbbell shape, which you will see how the shape is present. And here we can see there is stomata which is present. It is present in the lower surface. In case of dicots as well as in xerophytes, the stomata which is present, it is present in the lower region. So why it is present in the lower region in case of xerophytes? Because if it is present in the upper region, the stomata will be closed. If the stomata will be open during the daytime or nighttime, whenever the process is taking place, it will be open. So due to this, there will be more loss of water, more transpiration will take place. This is the reason why it is shifted, why this region is shifted into the lower surface. And in case of hydrophytes, this stomata which is present, it is present in the upper region, because in the lower region they are already present in the water, in the surface of the water. So from here the transpiration cannot take place, or the exchange of gases, it will be low, so it is present in the upper region. And in case of monocot plant, the stomata pore which is present, it can be in both the portion, like it can be in the lower region as well as it can be in the upper region. So here we can see the stomata. What is the requirement of the stomata? It will help in the gases exchange, transpiration, water balance, as well as plant temperature regulation. So here we were saying about the structure. In the guard shell, there is nucleus, as well as here the chloroplast is present. So what is the requirement of this chloroplast? This epidermal shell which is present, guard shell is only the epidermal shell which is having the chloroplast with it, and other epidermal shell, they are not having any chloroplast with it. You can see here that the subsidiary shell which is present, it is also a epidermal cell, but it is not having any chloroplast with it. So here the guard shell which is having the chloroplast, it will help in the photosynthesis, and due to this photosynthesis there will be formation of sugar, which is the glucose. Okay, so another thing which is present over here, it is the vacuole. Okay, and this guard shell which is present, the inner portion, this inner region which is present, it is a thick region. This is the thick region which is present, and the outer region is the thin region. And due to this thick and thin region, this is also helping in the opening of the stomata. It is also having the mechanism due to which the stomata is being opened. Okay, so here we can see that the in case of monocots, the stomata which is present here, it is of a dumbbell shape, and in case of dicots, it is having a guard shell of kidney shaped. Okay, and here the subsidiary shell, cellulose microfibril, or we can say the cellulose microfibrils are present here, okay, in the guard shells. So this is also the same diagram. Here the subsidiary shells are present and guard shell is present, which is having the chloroplast with it. So now let us see the some of the theories which is responsible for describing this two opening as well as closing of the stomata. The first is the starch sugar interconversion theory. Second one is the potassium ion exchange theory. So at first we will discuss how the starch sugar interconversion theory is taking place. So for this I acquire a diagram. So here we will discuss about that. So what happens in case of daytime? What happens in case of daytime? The light falls upon this cell due to which there is formation of glucose. Or during the daytime, what happens? During the daytime there is photosynthesis, and due to photosynthesis there is formation of glucose. Okay, this glucose which is present, it will be present in the guard shell. So due to this the solute will be more in the guard shell. Okay, as the solute is more, that means H2O will be low in the guard shell as compared to the outer environment, and here the outer environment is a subsidiary shell. Okay, so here due to more solute concentration in the guard shell, there will be end osmosis. That means intake of water. Okay, so from the guard, so from the subsidiary shell there will be intake of water inside the shell, inside the guard shell, due to which there will be turgor pressure inside the guard shell, and due to this turgor pressure the stomata will be opened. Okay, so in the stomata the inner region which was present, it was thick, and the outer region it was present, it was thin, and in between for connecting this there were cellular microfibrils which are present. When the water is entering inside the guard shell, there is some of the pressure, that means turgor pressure is taking place, and due to this turgor pressure there will be pressure in this thin portion first, due to which it was straight. You can see here that here the curve is little in when the stomata is closed, that it is not much curve, but due to the pressure of water this is bending. Here the swallowing is taking place, due to which there is pressure in this thin wall, and due to which the stomata is now opened. Okay, this is how the starch sugar interconversion theory is telling about the opening of the stomata. So this is in case of daytime. What happens in case of night time? In case of night time there is no photosynthesis taking place. So here there is no glucose. So as it is having no glucose, that means low solute concentration is present inside the guard shell. So water will be more inside the guard shell. So here there will be exosmosis, due to which the guard shell will be flaccid. And due to which there will be closure of the stomata. This is how the starch sugar interconversion theory describes about the opening of the stomata as well as closing of the stomata during the day and night condition. Okay, now next we will discuss about the potassium influx ion theory. So how this is being regulated. So what happens in case of dark light condition? We will see first. So here the guard shell which is present, it is helping in the day condition. Due to presence of light, glucose will be synthesized due to photosynthesis, and this glucose which is present, it will be converted into malic acid, and this is in the guard shell. This all things which we are discussing now, it is in the guard shell. And in the guard shell there are some channels, like, okay, first we will discuss about this malic acid. This malic acid, it will be dissociated into H plus and malate. Okay, it will be dissociated into H plus and malate. This, due to this high H plus inside, what happens, the pH will be low. Okay, so this H plus, as it is more inside the guard shell, it needs to be exported from the guard shell. So here for this export, it is having a pump in it. It requires, and it is H plus ATPase. It is H plus ATPase, and due to this H plus ATPase, H plus will be transferred out. Okay. So as much of the H plus is being transferred out, so here there will be low amount of H plus, due to which the pH will be more inside the cell. So for maintaining this, here there will be influx of K plus ion. Okay, so now it is being neutralized. Now it is being neutralized. Okay, so during this condition, there is being neutralized. So here there is, so for balancing this K plus and malate, so here the malate is present. Now this malate is converted into potassium malate. This malate is being now converted into potassium malate, which is being stored in the vacuole. Here it is being stored in the vacuole. Okay, so now for maintaining the osmolarity, here the chlorine ion which is present, it is being transferred, and it is being transferred into the vacuole. Here the chlorine ion which is present, it is being transferred into the vacuole. So here what happens, there is rise in osmotic potential. Osmotic potential is more now in the guard shell. So due to this osmotic potential, due to this, or we can tell due to the high solute potential inside the guard shell, there is endosmosis. So for it, the water is entering inside the guard shell from subsidiary shell. Okay, which is also known as endosmosis. Okay, and due to this endosmosis, there is turgor pressure. Turgor pressure is present inside the guard shell, and due to this turgor pressure, there is swelling of the guard shell, due to which it will be opened. Okay, there will be formation of pores. So this will be opened, and there will be formation of pore, which is known as opening of stomata. Okay, so this is how the regulation of opening of stomata is taking place by this theory, that is potassium ion influx theory. So what happens in case of dark condition? In case of dark condition, everything will be opposite. Like there will be no glucose formation. So as there is no glucose formation, there will be no malic acid. If there is no malic acid, there will be no H plus ion. So this H plus ATPase, which was open during this daytime, this will be closed. Like we can see here, like we can write here that this malic acid which was present, like see, in the night condition, there is no photosynthesis. No photosynthesis is taking place. So there is no glucose is present. As it is having no glucose, no malic acid. As it is no malic acid, no H plus or malate, so it does not require H plus ATPase pump. So this H plus ATPase pump is blocked. Okay, so K plus chlorine influx, it was also having chlorine influx channel, it was also blocked. Now the K plus influx channel is also blocked. As this guard shell which is present, it is not having much amount of solute. Now here the amount of solvent is more, due to which there will be exosmosis, that means giving of water to the subsidiary shell. Due to exosmosis, there will be flaccid condition of this guard shell, due to which there will be closing of the guard shell. In this way, opening and closing of the guard shell is taking place by the two kind of mechanism. One is the old mechanism, which is known as starch to sugar conversion theory, and the second one is the malate ion conversion theory. I hope this video is helpful for you and you have understood the mechanism, how it is taking place. In the next video, we will discuss what are the questions being asked in different type of exams related to this content. Thank you.